Welcome to the first of our two podcasts on the circulatory system. By the end of this podcast, we are going to be able to identify different regions of the blood vessel. There is a generic structure that we have to the blood vessels. The inner lining is going to be the tunica intima, tunic meaning coat or layer. Then we have the middle layer, the tunica media, and the outer layer, tunica adventitia, or at times we may refer to this as a serosa if it is in fact covered by mesentery. Secondly, we're going to classify arteries based on their histology. We have some that are very large and some that are much smaller. And then we're going to describe some of the regulatory structures and mechanisms responsible for the maintenance and the homeostatic balance of blood pressure. So as we look at this general structure of blood vessels, we know all arteries are defined as vessels that transport blood away from the heart. Now, generally speaking, the majority of these are going to be very highly infused with oxygen, but not all. The pulmonary circulations are going to be opposite of that rule. So arteries transport blood away from the heart. As we move in our arterial system to getting smaller and smaller and thinner walls, we will eventually arrive at capillaries that are only a single endothelial cell in layer, and this is where we're going to exchange materials, gases, nutrients, waste. Then we look at veins. All veins return blood to the heart. Again, some more generalities. Arteries are going to have thicker walls than veins, and veins are going to have larger diameters than arteries. These are some general rules that when we look at them, it will help you distinguish these histologically. In this illustration, we have one vessel here that we see and a second vessel. We can see a fairly thick wall. We have the endothelium on the inside. We have these layers of smooth muscle in the middle and then our connective tissue outside. This is going to be part of our arterial system. And again, as you look at the luminal diameter, it's not going to be much different, if at all. But now look at the luminal diameter versus the thinness of the middle portion especially. So this is part of our venous system. This is our arterial system. In an actual histological slide, once again, we can see the endothelium, and often these endothelial nuclei will budge out into the lumen. We have our smooth muscle layers, maybe about 3 or so. So this is going to be a small artery or a large arteriole. And then now we have a much larger luminal area and very little, if any, in the way of smooth muscle. So very thin wall, large lumen. This is going to be a small vein. Another image, and I think, again, from what we've talked about, you can distinguish artery from vein. Here we have the thick muscular wall of our artery. And here we have the very thin wall, again, fairly large luminal diameter. And often veins are going to have a collapsed look to them. And so this is the vein, and this is the artery. So now let's talk about the generalized structure of vessels. This will apply to both arteries and veins, and we're going to set up this generalized structure. Starting from the inside and moving out, we're going to describe the tunics or the layers. The tunica intima is that on the inside, and it consists of our endothelium, which classified is going to be our simple squamous epithelium. Then we also have the basement membrane on which that epithelium is attached. So here in our generalized diagram, we can see the different layers. Here we see the intact view, and these are cutaway sections. So our tunica intima, here is our endothelium, and we're going to have the basement membrane portion of the tunica intima. Now we're also going to have underlying connective tissue. Certainly we've got our lamina reticularis, and so we're going to have our collagens types 4. We're going to have collagens 5. We're going to have collagens 2. Sometimes we're going to have these here. Laminin, our cell adhesion molecule, and then we also have these molecules endothelin, which is a powerful vasoconstrictor, and we also have nitric oxide produced by our endothelial cells. Also, nitric oxide, a very potent vasodilator in this case. Now, we learned a little bit about von Willebrand's factor. This is going to be secreted and deposited into the basal lamina underlying the endothelial cells, and this is going to cause much more rapid coagulation and activation of platelets to form the platelet plug. We also see that we're going to have membrane-bound enzymes, and certainly in the lung, we have angiotensin-converting enzyme, ACE, and that's going to be responsible, recall, for converting angiotensin 1 to angiotensin 2, and this is one of the ways that we are going to regulate our blood pressure by regulating our fluid volume. Angiotensin 2, we're going to see, causes vasoconstriction, an increase in blood pressure, and that leads to the release of aldosterone. So in this case, we are going to produce aldosterone. We are going to be more thirsty. This is going to cause the body to resorb and retain more water. This is all in response, remember, to low blood volume and low blood pressure. So in our illustration, again, we have hypotension, low blood pressure, which is sensed through a decreased sodium delivery into our distal convoluted tubule. The kidney responds by producing renin. Renin is going to act and activate angiotensin-ogen. There's angiotensin 1. Angiotensin converting enzyme in the lung creates angiotensin 2 that's going to feed into all of these different areas. Here we have aldosterone. We're going to interact with the pituitary anti-diuretic hormone. And so that's going to cause our fluid retention in the kidneys to go up, increase in our blood volume, increase in blood pressure. Now, beneath the endothelium and the basement membrane, we have a reeler connective tissue. We may see some smooth muscle here, but not really anything we're going to identify at this point. But the bottom layer, the defining boundary between the tunica intima and the tunica media, is going to be the internal elastic lamina. So what we're going to see here is a fenestrated sheet of elastic fibers. So here in our generalized diagram, we can see our endothelium. We've got our basement membrane. They've cut away the green. Here we have our loose connective tissue, and then this is illustrating our internal elastic lamina, very nicely illustrating those fenestrations within that sheet. So in our histological diagram, often the endothelium, basement membrane, subendothelial connective tissue, and the internal elastic lamina is all tightly compacted together. And in this particular stain that really highlights that elastic lamina, that's really all we see is this line. This is all tunica media. This is smooth muscle with elastic sort of situated between. But here in a higher magnification, we can begin to distinguish the cells on the very outside, subendothelial connective tissue, and then here you can see that very dark line. That is our internal elastic lamina. That's going to define the boundary between the intima, which is this area, and the tunica media, which is this area. Now the role of the fenestrations in this lamina, this very porous layer, it's to allow diffusion of material. I think you can envision if we are having to diffuse materials from the lumen where we have our blood cells, that's only going to diffuse as deeply as sort of the density of the tissue. So those fenestrations are going to allow materials to more easily diffuse through the tunica intima and get deeper into the tunica media. Now the tunica media, this is going to be the thickest layer of most of our blood vessels. Certainly this applies to our arteries, but in many cases it's also going to apply to our veins. This is going to be comprised of circularly arranged smooth muscle, and I think that makes sense if you think about it. These smooth muscles are going to be able to contract, to constrict the lumen of the blood vessel, but dilate as well, and as they do, expand that luminal diameter to regulate blood pressure and blood flow. So in addition to smooth muscle, we see elastic fibers. Type 2 collagen and proteoglycans are going to be a part of this. So type 2 collagen's beginning to come into play now, much more so than we saw in just our tissue histology with cartilage. But we see this also within the circulatory system, and during cardiac development, we'll see some of type 2 collagen as well. Now, the tunica media is defined by the area immediately underlying the internal elastic lamina, and it extends downward through these muscle layers all the way and includes the external elastic lamina. Once again, this is going to be a sheet of elastin that's fenestrated, separating the tunica media from the tunica adventitia. Here we see we've left off with our internal elastic lamina. Here we have our tunica media with our muscular layer, and then this we see is our external elastic lamina. It looks much the same as the internal elastic. The only difference is the location. Once again, we're looking at an artery. We have our internal elastic lamina, which really is going to be defining our tunica intima boundaries. And again, in this one, we can't really distinguish endothelium, subendothelial connective tissue, and our internal elastic lamina. And I think in this particular stain, that real pale line is going to be our internal elastic lamina. Here we see our smooth muscle, that nice flowing nature to what we see. And then we have another boundary, very sharp distinction, and this dark edge that we see, that's going to be the external elastic lamina, and that's going to define the boundary between the tunica media and our underlying tunica adventitia. So the third and the deepest tunic is the adventitia. This is going to be the outermost layer, and what we're going to see, again, more loose in the way of the space between. We've got a lot of fibroblast, a lot of collagen, not college, collagen type 1, and elastic fibers are going to be abundant in this tissue as well. So here we have the tunica intima, and we see that extends with our endothelium that we can see, subendothelial membrane, subendothelial connective tissue, and then our internal elastic lamina is running along about here. In this location, it doesn't go that deep. I'm not sure why they put that mark there. At this point, that would be correct. But then we have the tunica media. Here we have our external elastic lamina, and then our tunica adventitia. This is going to look much more like loose or possibly dense, irregular connective tissue with our collagen fibers, elastic fibers as well. Now, it's within the adventitia that we have blood vessels that supply blood vessels. I know that sounds a little bit anti-intuitive, but our internal and external elastic lamina, they're going to have fenestrations that are going to allow for diffusion, but as the thickness of the vessel gets bigger and bigger, that diffusion is not going to make it all the way down. So for our larger vessels, we're going to need their own blood supply to nourish the deeper layers in the media and the adventitia. And so these blood vessels are present in the adventitia, and we refer to them collectively as the vasa vasorum. So once again, here we have our vessel, the tunica intima, very thick tunica media, and then we get down into our tunica adventitia. We can see some adipose tissue down here, and now we see cross sections through fairly large blood vessels, but you can certainly see not as large as this one. And so this would be our vasa vasorum. Now, with our blood vessels, they are going to have an intrinsic response to blood pressure. It's sort of an intrinsic vasoconstriction to resist blood pressure, but we do have nervous system control. Our autonomic nervous system will be able to do this. So norepinephrine, for instance, it is going to diffuse into the smooth muscle, and it is going to cause a constriction, whereas acetylcholine, so our cholinergic nerves, part of the parasympathetic system, that's going to lead to vasodilation, especially in our skeletal muscles. And remember, in a parasympathetic response, that's our rest and digest mode. Okay, now that we have our generalized structure, we're going to look at how these layers differ, and that allows us to then classify our blood vessels. And we're going to start with arteries. And the largest of our arteries in thickness of wall and luminal diameter are going to be our elastic arteries, and we refer to these as the conducting arteries. And so we're going to see the aorta common carotid, subclavian, the iliacs, and the pulmonary trunk. These are going to be examples of our conducting arteries, and they are of the elastic type. So as we look at the three layers and how they differ, our tunica intima is going to be extremely fenestrated to the point that it is almost incomplete. So we're going to allow a tremendous amount of diffusion to happen here. As we look at the media, we are going to have alternating layers of smooth muscle and elastic sheets, and again, these are going to be fenestrated, and we may have as many as 40 to 70 of these alternating layers. So we're going to see a very thick media, as we mentioned before. That's usually the thickest of all the layers. The external elastic lamina is going to be extremely thin, very porous, again, to allow as much diffusion as possible, not necessarily from the luminal diameter, but from the vasa vasorum that is down in our tunica adventitia. So here we have a large vessel. We have the lumen, we have our tunica intima, and here we have our alternating layers of smooth muscle and elastic lamina. This is stained for elastin, so we see the dark layers going back and forth, many of these. So this would be an example of one of our elastic arteries. Here's the adventitia here. Here's the intima, and an extremely thick layer, the tunica media. Now, as we decrease in our luminal diameter, we're also going to drop off from all of our different, so many layers of our muscle. These are still going to be fairly thick, but these muscular arteries aren't going to have as thick a media as we saw with the elastic arteries. So you're going to have to kind of keep that in mind. These muscular arteries are going to be of the distributing type, and most of our named arteries, except for the ones we did with the conducting system, most of those are going to be part of this distributing arterial system. So these named arteries, even some quite small, maybe 100 millimeters in diameter, 100 nanometers in diameter, those may in fact still be muscular arteries. So here for our muscular artery, very distinguishing features, a thick, very prominent internal elastic lamina. And the media, this is where instead of seeing 70 layers, we're going to see anywhere from 40 layers down to about 3 or 4. And certainly as the diameter of these muscular arteries decrease, so will the layers of smooth muscle. Also, very thick external elastic lamina that's really going to define those boundaries. We saw those before as we were looking at the media. That's what's going to be popping up here. Now, the adventitia does have some vasa vasorum, but they won't be as easily recognized. They're not going to be as large because as we have fewer layers, as our fenestrations are quite porous in our internal and external elastic lamina, diffusion will supply most of the layers of these vessels, and the vasa vasorum aren't as critical to be present. So here's our image that we have seen before, but clearly we have an extremely prominent internal elastic lamina, external elastic lamina. Now this stain is for elastin, and we don't see all of the elastic lamina and sheets. We have elastic fibers, and in fact when you look at this inset magnification, you can see one of those elastic lamina. But most of what we're seeing here in the media is smooth muscle rather than our elastic lamina. Here we see another muscular artery stained for elastin, and here in our adventitia we see a lot of elastin stain. But clearly we've got our internal elastic lamina, external elastic lamina with our smooth muscle layers between, with some occurrences of elastic. But again, here we're going to have certainly more than 3 layers of smooth muscle, less than 40, so we are in the muscular artery range. And the internal and external elastic lamina allows us to distinguish that from an elastic artery. Now, as we continue to get smaller in diameter, we get to the arterioles. And these are the terminal arterial blood vessels. This is just immediately before we get to our capillaries. And with our arterioles, the diameter is approximately going to equal the width of the vessel. Now with our intima, we may not see the internal elastic lamina. We are not going to have an external elastic lamina at all. And so the media, we're going to be counting layers of smooth muscle, anywhere from 1 to 3 layers of smooth muscle. And then, of course, we're going to have somewhat of this fibroelastic connective tissue of the adventitia, and it's going to be much thinner than we've seen with our larger muscular and the largest elastic arteries. Now, this is a transmission electron micrograph, and in this particular instance, we can see the lumen of a vessel. We have our endothelium. We have our subendothelial connective tissue here. We don't have the internal elastic lamina, so this is all we have in the way of the media. And here we have one, and we might have another smooth muscle cell somewhere, maybe beginning over here, but for all intents and purposes, one smooth muscle layer thick. And then we have our collagen fibrils, and you can see we've got individual collagen running through here. So that's the thickness of the entire wall of that vessel, and this in fact is an arteriole. Here we can see some other examples. Once again, here we see our diameter of the lumen. We have our endothelium. Here we have 1, 2, 3-ish layers thick. We have our adventitia on the outside, and so this is going to be an arteriole. Here we have another transmission electron micrograph showing our endothelium, basement membrane very well defined, underlying connective tissue, and then our smooth muscle cells, one layer. This is also going to be an arteriole. Now, in this case, our lumen seems to be getting a little bit larger here. So we might be thinking a small vein, but still look at the thickness of this wall versus the luminal diameter. And it's possible we're cutting this in longitudinal section because you can see this is somewhat oval shaped. So we're still going to go with arteriole in this particular illustration. Now, a different type of arteriole, it's basically going to have the same 1 to 3 layers. You're going to see your endothelium. You're going to see basement membrane. No internal, external elastic lamina. But in this case, these are immediately before we get to the capillaries. And so really the only difference between these and the smallest of our arterioles we talked about before is their location. And these are going to be the pre-capillary sphincters. These bands of smooth muscle that make up the media, they are going to regulate the blood pressure of the blood entering into the capillary bed to ensure that it's not under such high pressure that it would damage the underlying capillaries. So once again, we have our arteriole. We can see our endothelium, basement membrane, and here we have our smooth muscle on the outside, one layer, so we're talking about an arteriole. And here in the scanning electron micrograph, you can see how these smooth muscle layers are wrapping around this blood vessel. It's difficult for us to see how many of these we have, but I believe this is one smooth muscle layer. I believe this is an arteriole that we're looking at, but that gives you an example of these circularly arranged smooth muscle cells around those blood vessels. Now, we mentioned we're going to regulate blood pressure, and this is a bit of a review from what we have talked about in physiology, so we're going to go through this fairly quickly. The carotid sinus, this is going to be located in the internal carotid artery just after it branches off or bifurcates from the common carotid, and it is in fact a baroreceptor. It's going to be sensitive to those changes in blood pressure. We're going to send those fibers, sensory fibers, through the glossopharyngeal nerve. It is a mixed nerve. It's both motor and sensory. We're going to send that information back to the brain about the blood pressure that we have coursing through these areas. So here we can see in this diagram where we're going to have the carotid sinus in this case. And so here we have the internal carotid coming off of the common carotid. So it's going to be right in this location where we're going to have the carotid sinus. Now, the other type of receptor we have is the carotid body. And this is going to be in a fairly similar location, but instead of being a baroreceptor, this is a chemoreceptor. And it's going to be sensing pH, which is going to basically be a reflection of the carbon dioxide levels of the blood. And in the carotid body, we're going to have two types of cells. We've got the glomus, the type 1 cells, and we have the sheath type 2 cells. So these glomus cells are going to be the ones centralized within the carotid body. And when we look at neurons, for instance, we know we have presynaptic cells and we can have postsynaptic cells. Well, in this instance, these cells seem similar to presynaptic because they're going to secrete catecholamines. And they're going to secrete these in response to our pH levels of the blood. Then we look at the Schist cells. These are basically going to be the ones that cover over, and they're going to form the capsule then around the glomus cells, and they're going to also cover over the axons, which are entering into the carotid body. So in our illustration, we have the carotid body. The carotid sinus is going to be, again, sort of in this direction. They're calling this the carotid sinus nerve, sensing, again, those pH changes. Here we have the Schist cells on the outside, the glomus cells on the inside, and here we have a synapse. So this is almost going to be like our postsynaptic membrane, and what we're going to be secreting here, we're secreting acetylcholine, but you can see we're also secreting norepinephrine, they call it noradrenaline in this case, dopamine, these are our catecholamines. Other substances are going to be detected by this axon taking information to the brain to tell the brain how to respond to these altering changes in pH and thus changes in CO2 level or respiration state of the body. The aortic body, this is going to be located around the aortic arch, and what we're going to see is it's going to have a similar function to the carotid body. It is going to be a baroreceptor, and so it's going to assist in the identification of changes in blood pressure. Now, all of this information coming from the carotid sinus, carotid body, the aortic sinus, that is going to come to the vasomotor center of the brain. And this vasomotor center is going to take all of this incoming information, and it is going to have a certain given state of vascular contraction called vascular tone. And so this is going to stimulate vasomotor nerves to cause vasoconstriction, and that's going to be a sympathetic response, whereas parasympathetic fibers will be activated to release acetylcholine to stimulate vasodilation during that response. Now, we can also see acetylcholine, when it interacts with endothelial cells, this is going to cause the release of nitric oxide. And that release of nitric oxide is going to cause vasodilation. It's going to cause the smooth muscle cells to relax. We also see, however, that in certain situations, if you are severely injured and you are hemorrhaging, then that's going to lead to the release of ADH. And in this case, ADH, antidiuretic hormone, in addition to conserving water, because you're losing an extreme amount of blood volume, it's also going to be a potent vasoconstrictor to try to restrict the blood being lost systemically, but also from the area of the hemorrhage. This is a flow chart just trying to illustrate you have your changes in arterial pressure, and we're going to sense those through both our carotid and aortic bodies and sinuses. That's going to feed back to the vasomotor center of the brain, number 4. We also have the vagal center, and these are going to go out to our arteries, our vascular supply through the sympathetic nerves. It's going to impact the heart, which is always going to feed back on our arterial pressure in a negative feedback mode that we learned about in physiology. And all of this ties together our ability to detect changes in our arterial pressure and respond to those changes on almost a steady state, constant minute by minute process to make sure we regulate that within homeostatic levels.